Okay, welcome to video number 57 of the Diaries of a Coronavirologist YouTube channel. Today is the 7th of April, and for anyone new around here, I'm Stuart. I'm a postdoc researcher who's been studying coronaviruses since 2016, and that makes me a coronavirologist. So welcome to part 2 of my two-part video series looking at how Neanderthal DNA, or DNA inherited from Neanderthals, is impacting severity of COVID-19 disease. There's a link to last week's video down in the description for anyone who wants to go back to it, for anyone who hasn't seen it. But just as a brief recap, I was talking about how DNA has been inherited from Neanderthals, talking about out of Africa and admixture between Neanderthals and modern day, or what are now modern day humans, but at the time were ancient Homo sapiens. And then how there's two studies which have looked at some of that Neanderthal inherited genetic variation and how that impacts COVID-19 disease severity. I focused predominantly last week on a paper that looked at part of chromosome 3 and found that that was associated with an increased risk of being hospitalised if infected by SARS-2. What the exact role of the genes in that area of chromosome 3 are is a bit unclear. And at the end of last week's video, I alluded to another study by the same authors which looked at part of chromosome 12 and found that this is associated with an increased protection from being hospitalised by COVID-19, so a decreased risk of severe disease. That region on chromosome 12 is better understood, and it encodes a set of genes which make sense to be associated with protection from viral infection. And those are what I want to talk about tonight. Both of those papers are linked down in the description for this video for anyone who wants to go and look at the original research. The genes on chromosome 12 are known as OAS genes. So in this video, I want to take a look at what OAS genes and OAS proteins are and talk about how they can activate another protein in the cell which is known as RNaseL. So I'll talk about what that is as well. Now these two protein groups together make a good antiviral system. They help defend cells against viral invaders, such as SARS-2, which is why they may be involved with protection against severe COVID-19. However, they can also have some detrimental impacts. So this sets us up with a little interesting evolutionary trade-off where they can be good when there's viruses, but they may not be so good when there's not. So I'll look at that evolutionary idea by talking about the Neanderthals and how that's impacted evolution and the positive selection that's led to the development of these Neanderthal versions, which may be protecting against severe COVID-19. And I'll also then close out by looking at the OAS genes in monkeys and non-human primates, just to flesh out this idea about evolution more, arriving at a point whereby I'm basically saying that viruses have driven a huge amount of human evolution. So that's where I'm going to go with this, while talking about severe COVID-19 and genes associated with it, of course. As I say, the genes that are associated with protection from severe COVID-19, for which there are genetic variants inherited from Neanderthals, are called OAS genes. And there are 3 of them in this region on chromosome 12, which are OAS1, 2 and 3. OAS stands for oligoadenylate synthase. I'm just going to call it OAS throughout the course of this video. And the naming means the synthase, it's an enzyme, it produces oligoadenylate. OAS proteins play a very important role in our cells for protecting against viral infection. They form part of the antiviral response within our cells. We're working in concert with another protein called RNaseL. So an RNase is a protein that degrades RNA, and here the L stands for latent, which I will get to momentarily. But before I do that, I want to just take a little step back and have a little think about how viruses replicate within a cell. Obviously, a virus goes in, it infects the cell, and it turns it into a factory to produce more and more copies of itself, which can be damaging to the cell, which is why our cells have evolved mechanisms to counteract that. So if we take coronaviruses, SARS-2 specifically, the virus that causes COVID-19, these are positive sense RNA viruses with a single strand of RNA that makes up their genome. However, while that genome is just a single strand of RNA, it's possible to also have double-stranded RNA. So if you can think about this in the same way as the human genome. So the human genome is double-stranded DNA. If you think of the double helix structure, that is two strands of DNA wound together to form a double helix. Now the way that that structure is held together is through interactions between the building blocks of DNA, the nucleotides, which are denoted as A, T, C, and G. So A and T bind to each other and C and G bind together, and that's what holds this double helix together. The same can be true for RNA. So while the coronavirus genome is single-stranded RNA, you can also have double-stranded RNA, which forms through interaction of the RNA nucleotides. In the context of RNA, they're called A, U, C, and G, so there's no T in RNA, but the same applies. So A binds to U, C binds to G. To give an idea of this and to give an example, if you imagine a stretch of RNA that reads A, C, C, and then if you have another strand of RNA which reads U, G, G, those two will bind very nicely and form double-stranded RNA. When viruses, such as coronaviruses, replicate within a cell, they actually produce quite a lot of double-stranded RNA in this kind of manner through this complementary pairing. In order to make more copies of their genomic RNA, the coronaviruses produce a complementary strand. So if the genome was AACC, they'd make a strand which reads UUGG, and then from that UUGG, just make the complementary paired over and over and over and over again to replicate their genome. That's essentially how a virus makes more copies of its genome, by using this matching complementary template. Obviously, I've simplified things here by talking about only 4 nucleotides. The coronavirus genome is around 30,000, but you get the point I'm getting at. However, because both of those different orientations of the RNA, those different complementary strands, are produced, this means double-stranded RNA is produced during the course of a coronavirus infecting a cell and replicating. Now the thing is, our cells don't really produce much in the way of double-stranded RNA. So the presence of double-stranded RNA in a cell is actually a really good warning sign for a cell to realize it's been infected by something foreign, something that could be bad, something dangerous. So that's almost like an alarm going off in your house, like an intruder alarm alerting you or neighbors or someone to the fact that there is something wrong. And one of the ways that the double-stranded RNA is detected is through OAS. So this brings me back to the OAS proteins. They can bind to this double-stranded RNA, realize that this is out of place, this isn't something that should be here, and oh no, there's a virus infection, we need to do something. And that something is to produce 2,5-oligoadenylate, to give it its proper name, 2,5-A. So oligoadenylate synthase produces 2,5-oligoadenylate. And what that 2,5-A does is it activates RNaseL. So again, bringing back in this protein that degrades RNA, and its full name is RNase latent. It's latent until it becomes activated by 2,5-oligoadenylate produced by OAS. And what RNaseL does is it destroys RNA. It destroys all of the viral RNA within a cell, which is a really good way to fight back against a viral infection. So that cell has a countermeasure to try and fight back against the coronavirus that's infected it. However, proteins aren't all that clever, and RNaseL is very good at degrading RNA, and it doesn't just degrade viral RNA. It goes berserk and it takes down all of the RNA within a cell, which essentially means that cell's going to die. Obviously, it's good to remove the viral RNA because that's how you stop a viral infection, that's how you stop that cell producing more viruses, but in the process that cell will die because of RNaseL destroying all of the cellular RNA that's needed for that cell to maintain functioning. So it's kind of a scorched earth tactic. There's a virus here, let's just destroy this cell. We don't want any more virus coming out, here's a way we can try and stop it. So in a sense, the virus may win the battle, but the organism, the multicellular organism, may win the war. If you stop that virus spreading within the human body, that can limit disease severity, and therefore people don't die from severe COVID-19. So from all of that build-up of talking about what OAS proteins are and what RNaseL is and how the two interact, hopefully you're starting to see why having more active OAS or more active RNaseL may be beneficial, and why maybe they could be associated with a decreased chance of severe COVID-19 or any other disease. However, there is a little kink to this story, because while I say that there isn't much in the way of double-strand RNA within a human cell, and therefore it acts as a good warning signal for a virus to trigger this mechanism of destroying that cell in the hopes of stopping the infection of the whole organism, our cells do produce some double-strand RNA, and therefore you can get some activation of the OAS RNaseL axis, even when there's no virus. And that can of course have some detrimental impacts. It's been shown in studies in mice, for example, that if you remove RNaseL or OAS genes and you keep those mice in a sterile environment, they live for longer than mice who have active OAS and RNaseL genes. So it suggests there is a trade-off between the antiviral actions of these proteins and the destructive, damaging actions that they can have if they're not well controlled. So that then brings me on to talking about the Neanderthal OAS genes and their link to protection against severe COVID-19. So as I was saying, it seems like a very good way to stop replication of a virus and therefore stop disease is to have more active OAS. And indeed, the Neanderthal versions of the OAS genes have been seen to be more active. So in the paper that I've put a link to in the description, published in the journal PNAS, they found that the Neanderthal versions of OAS on chromosome 12 appear to protect against hospitalisation from COVID-19 with about a 22% reduced risk of being hospitalised. And this is really interesting to see, because it's all well and good studying how these proteins function in a cell. That's the kind of stuff I love doing, that's the kind of research I do. But it's nice to see that translate out into a wider situation of humans being protected from disease. Now, what's also interesting about these genes is it's not just SARS-2 that they appear to protect against. They also seem to protect against West Nile virus, hepatitis C virus, and there was even a suggestion they may have protected against SARS-1. So now that we have more data because of SARS-2 infecting significantly more people than SARS-1, it looks like they're also actively protecting against this coronavirus. And it wouldn't surprise me if it's actively protective against other coronaviruses too. But now let's try and bring some of this together, some of these ideas. I've been talking about how the OAS genes are good in the context of a viral infection because they seem to protect against severe disease. But they also might have this negative effect of being activated when they shouldn't be and causing damage to the cells and therefore potentially lowering life expectancy, as seen in the mice, for example. So that raises the question of why did the Neanderthal versions of the OAS genes emerge? Why were they evolutionarily selected for? And the idea is that when the Neanderthal populations left Africa and ventured out into Europe or Asia, wherever it was they went, they may have come into contact with a virus which selected for more active OAS function. I'd speculate the virus probably came from an animal that they were hunting that didn't live in Africa. So as they go out and find this new environment, these new plains to roam, and they come into contact with new life, they come into contact with new viruses, and that puts an evolutionary pressure on them. So it seems that the more active forms of OAS that the Neanderthal variants are may have been selected for by a virus. As ancient Homo sapiens then also left Africa and went out into Europe and Asia, they would have encountered the same evolutionary pressures in that environment. Therefore, in the interbreeding that occurred between Homo sapiens and Neanderthals, it became a selective advantage for any that got these OAS variants which were more active, and they continued to then be selected for. And this is all backed up by the fact that in sub-Saharan African very close genetic relatives, or in sub-Saharan African ancient Homo sapiens, there is very little, if any, incidence of these Neanderthal versions of the OAS genes. However, in Eurasian heritage people, there's about 25 to 30% who have the Neanderthal versions. In the paper, the authors even speculate that this may be playing a contributing factor to why certain populations are more likely to die from COVID-19 than others. Now, the identity of such a virus I'm speculating about here that may have driven the evolution of the Neanderthal versions of the OAS genes will probably stay a mystery. We'll probably never find it because there's probably no remains of it. However, I find it fascinating to see that that kind of idea is playing out in front of our eyes now, just in a much more modern context where it's not going to be quite the same. So these genes are associated with a decreased likelihood of being hospitalised by COVID-19. Obviously in the modern world, that's okay. COVID-19 doesn't kill a huge number of people. Hospitalised people don't all die. There's good treatments and things like that. But in the ancient plains of Europe or Asia, if you're getting laid down by a viral infection for a week or two, or you require modern day hospitalisation, that's probably not going to be very good for your survival. And that's how evolution works. You select survival of the fittest. Those who developed these mutations in OAS that gave more active OAS function, they were selected for and they survived. Homo sapiens, who then came in contact with them, had the same selection pressures and went through the same kind of thing. And so that is viruses driving human evolution. And now, just to close out these ideas about evolution and viruses and the OAS genes, I want to take a little look at monkeys. Because there's another really cool paper, which I'll put a link to down in the description, which looks at this. So obviously monkeys and primates are closely related to humans and Neanderthals. It's all in the same part of the evolutionary tree. And what's been found is that certain species have lost function of the OAS genes. For example, we humans obviously have functional OAS genes. So do chimpanzees and orangutans. However, gorillas seem to have developed a mutation whereby they lose function of the OAS genes. And to go slightly further evolutionarily connected, tamarin monkeys have also lost the function of their OAS genes. So this is the idea that there is an evolutionary trade-off. The OAS genes and RNaseL, that axis of those proteins, really great and really beneficial in the context of coming into contact with viruses that they help protect against. But if you're not coming into contact with any viruses that they're beneficial for, they are detrimental and therefore they can be evolved away from. It's also interesting to think about what other defenses the likes of gorillas or tamarin monkeys may have developed because they may have come into contact with other viruses. So the point I'm trying to get to and convey to you in this YouTube video is viruses have driven human evolution and evolution of all life on the planet, and that actually the defenses we've evolved to counteract viral infection, which are beneficial when we're in contact with viruses, may not be so beneficial when not in contact with these viruses. And that's just something I find very fascinating and why I really enjoy what I do. And with all that said, thank you for watching. If you enjoyed the video, please drop a like for the YouTube metrics. Please leave any questions and comments down below and I will do my best to get to them as time allows. And if you're new around here and you found the video interesting or useful, please hit the subscribe button. And as always, please stay safe, wash your hands, wear a mask and keep calm and carry on. We will get through this pandemic.